This is ChainMind, the decentralized AI network built for everyone. Just search ChainMind on Google, sign up, download the app, and you're in. That's it. You've joined the network. Run it on any device, phone, laptop, or server. Why ChainMind? Fully decentralized, fully yours.
What if the key to stopping Alzheimer's isn't hidden in the brain at all, but tucked away in the bacteria living in our gut? Imagine a tiny molecule, born in the colon, slipping past the body's most guarded border and silently nudging neurons toward the plaques and tangles that define dementia. This is not a sci-fi plot twist, it is the emerging reality of gut-derived neurodisruptor, a metabolite that may rewrite everything we thought we knew about Alzheimer's onset, detection, and treatment. The story begins with the gut-brain axis, a concept that was once a fringe curiosity in neuroscience labs and is now a cornerstone of modern biology. For decades, scientists knew that the trillions of microbes in our intestines help us digest food, train our immune system, and even influence mood. Epidemiologists started to notice patterns. Diets rich in fiber and fermented foods correlated with lower rates of depression and cognitive decline, while high-fat, low-fiber diets seem to accelerate brain aging. Over the last 5 years, researchers built on those clues, showing that microbial metabolites can cross the intestinal wall, enter the bloodstream, and interact with neural pathways. The breakthrough came when a team of biochemists isolated a single, previously overlooked compound produced by a subset of Firmicutes bacteria. They named it gut-derived neurodisruptor, or GND, and discovered that this molecule acts like a molecular Trojan horse, breaching the blood-brain barrier and setting off a cascade that mirrors the earliest stages of Alzheimer's disease. In the laboratory, the evidence was striking. Using cultured brain endothelial cells, the scientists added nanomolar concentrations of GND and watched the tight junction proteins, CLAUDIN5 and occludin, fade by nearly 40% within a single day. Those proteins are the mortar that holds the blood-brain barrier together, preventing toxins and immune cells from flooding the brain's delicate environment. When the barrier weakens, the brain becomes vulnerable. To see whether this effect translated to a living organism, the researchers gave mice a low-dose oral regimen of GND for several weeks. Within weeks, the animals displayed measurable leakage of Evans blue dye into the brain tissue, a classic sign that the barrier had been compromised. Magnetic resonance imaging confirmed microleaks in regions critical for memory, such as the hippocampus. Behavioral testing revealed that the GND-treated mice struggled with spatial navigation tasks that healthy mice mastered effortlessly, a mirror of mild cognitive impairment, the clinical precursor to full-blown Alzheimer's. Post-mortem analyses of human brains added a sobering human dimension. Cerebrospinal fluid from patients diagnosed with Alzheimer's contained significantly higher levels of GND than fluid from age-matched controls. The convergence of cell culture, animal models, and human tissue painted a compelling picture. GND is not just a byproduct of gut bacteria, it is an active participant in neurodegeneration. Understanding how GND fuels disease requires unpacking 3 intertwined mechanisms. First, the barrier disruption itself creates microscopic fissures that let peripheral inflammatory cytokines, interleukin-6, tumor necrosis factor alpha, sneak into the brain. Those molecules are normally kept at bay, but once inside they ignite microglia, the brain's resident immune cells. In their activated state, microglia shift from vigilant caretakers to aggressive warriors, releasing reactive oxygen species and nitric oxide. These free radicals batter neuronal membranes, eroding synaptic connections and impairing the brain's communication network. Second, the inflammatory environment primes the brain for proteinopathy. Amyloid beta peptides, which already have a tendency to aggregate, become more prone to misfolding under oxidative stress, while tau proteins lose their stabilizing phosphorylation patterns and tangle into neurofibrillary knots. The third mechanism is a feedback loop. As plaques and tangles accumulate, they further impair vascular integrity, widening the breach in the blood-brain barrier and inviting even more inflammatory mediators. This self-reinforcing spiral can explain why some individuals experience rapid cognitive decline despite modest genetic risk factors like APOE4. It also suggests that intervening at the gut level, before the barrier even begins to crack, could halt the entire cascade. If the gut is the launchpad, then the most powerful weapons may be early detection and preventive modulation. Traditional Alzheimer's diagnostics rely on expensive PET scans or invasive lumbar punctures to measure amyloid and tau levels in the cerebrospinal fluid. Those approaches are impractical for routine screening in a population of millions. By contrast, a stool-based metabolomic assay could identify elevated GND or its bacterial precursors long before the blood-brain barrier shows any signs of leakage. Pair that with a simple blood test for markers of barrier integrity, proteins like S100B or matrix metalloproteinase 9, and clinicians could generate a composite risk score in a single office visit. Imagine a primary care physician ordering a stool sample, receiving a rapid readout of GND concentration, and instantly seeing a risk flag that prompts dietary counseling, probiotic recommendations, or enrollment in a preventive trial. Early studies are already testing such pipelines. A pilot cohort in Italy used high-throughput mass spectrometry to profile stool metabolites, and participants with the highest GND levels showed a 2.3-fold increase in progression from mild cognitive impairment to Alzheimer's over 3 years, compared to those with low levels. The preventive angle opens a whole new toolbox of interventions. Dietary fibers that nourish beneficial bacteria, prebiotic compounds that suppress GND-producing firmicutes, and targeted antibiotics that selectively prune those strains are all under investigation. One small clinical trial in Japan introduced a probiotic blend rich in Bifidobacterium adolescentis, a species that competes with GND-producing microbes for the same carbohydrate substrates. After 12 weeks, participants displayed a 30% reduction in stool GND concentration and modest improvements in memory recall tests. Another approach involves engineered microbes designed to metabolize GND into inert byproducts, essentially turning the gut into a detox factory. While these strategies are still in their infancy, they illustrate a paradigm shift. Instead of trying to clear amyloid plaques after they have formed, we may be able to prevent the very conditions that allow plaques to emerge. Turning these promising ideas into clinical reality, however, demands a data infrastructure that can handle massive, heterogeneous datasets while respecting patient privacy. Researchers must integrate metagenomic sequencing of stool samples, metabolomic profiles, proteomic snapshots of blood biomarkers, neuroimaging data, and longitudinal cognitive assessments, all of which can amount to terabytes of information spread across hospitals, research labs, and even patients' own devices. Traditional centralized cloud platforms stumble on 3 fronts. Latency, because data must travel long distances for processing. Regulatory hurdles. As health data is subject to strict privacy laws like GDPR and HIPAA. And cost, since transferring and storing such volumes can quickly become prohibitive. This is where a decentralized AI inference network like ChainMind can make a decisive difference. ChainMind flips the model on its head by moving computation to the edge. Imagine a network of participating sites, university research hospitals, community clinics, and even home-based health devices, each holding its own raw data locally. a graph neural network model designed to predict blood-brain barrier permeability based on microbial gene expression and plasma cytokine levels is uploaded to the network, but the raw data never leaves its source. Instead, encrypted model weights travel to each node, the inference runs locally, and only the aggregated risk score is shared back, protected by homomorphic encryption. This architecture offers three concrete advantages for gut-brain Alzheimer's research. First, privacy is preserved by design. Patients retain ownership of their stool and blood data, and only anonymized, encrypted outputs circulate. Second, bandwidth usage drops dramatically because only model parameters, not gigabytes of raw sequencing reads, traverse the network. Third, the system delivers low-latency insights. A clinician can receive a real-time risk assessment during a routine visit, enabling immediate discussion of preventive measures. The incentive structure of ChainMind further accelerates collaboration. Researchers who contribute high-quality datasets earn tokens that can be used to access premium models or to fund additional compute cycles. Model developers can monetize their algorithms by licensing them to the network, ensuring that breakthroughs are shared rather than siloed. Because the network is governed by smart contracts, every contribution is auditable, and rewards are distributed automatically according to pre-agreed rules. This creates a virtuous cycle. More data improves model accuracy, which in turn attracts more participants, expanding the pool of diverse, multi-ethnic samples necessary to validate GND as a universal biomarker. Of course, no transformative technology is without challenges. Standardizing stool collection remains a thorny issue. Differences in storage temperature, time to processing, and sequencing platforms can introduce noise that confounds model training. ChainMind addresses this by hosting community-driven standards committees that publish best practice protocols and certify compliant laboratories. Interpretability is another hurdle. Clinicians need to understand why a model flags a patient as high risk, not just see a black box score. To meet this demand, ChainMind integrates explainable AI layers that highlight which microbial pathways or cytokine levels contributed most to the prediction, providing a narrative that clinicians can translate into actionable advice. Equity of access also requires careful attention. While high-end research hospitals may have powerful GPUs, low-resource clinics might only possess modest CPUs. The network's token economy can subsidize compute for underserved sites, and lightweight model versions can run on smartphones, ensuring that the benefits of cutting-edge AI are not confined to wealthy institutions. Finally, regulatory alignment is essential. Any diagnostic tool that influences patient care must undergo rigorous validation under FDA or EMA frameworks. ChainMind's governance layer records every model version, training dataset, and inference event, creating an immutable audit trail that simplifies compliance reporting. Looking ahead, the gut-derived metabolite itself could become a therapeutic target, and AI will be the engine driving each step. In silico screening of millions of small molecule libraries can identify inhibitors that block the bacterial enzyme responsible for GND synthesis, all without moving proprietary compound data onto a central server. Decentralized inference can evaluate these candidates across diverse microbial genomes, accelerating the discovery of broad-spectrum inhibitors. Personalized probiotic design is another frontier. Machine learning pipelines can simulate how a patient's existing microbiome will respond to introduced bacterial strains, predicting whether a probiotic will outcompete GND producers or inadvertently create new imbalances. Clinical trials for such interventions can be optimized by using real-time GND measurements to stratify participants, enriching the study cohort with individuals most likely to benefit and thereby reducing the number of participants needed for statistical significance. In this way, the entire drug development pipeline, from target identification to patient selection, becomes more efficient, less costly, and more patient-centric. The convergence of a gut-derived molecule that can breach the brain's defenses, a suite of preventive strategies that act far upstream, and a decentralized AI infrastructure that democratizes data analysis paints a hopeful picture for Alzheimer's research. It suggests that the disease may not be an inevitable consequence of aging but a modifiable condition that can be intercepted at its very origin. Yet the journey from laboratory discovery to bedside application will require coordinated effort across disciplines: microbiologists, neurologists, data scientists, ethicists, and policymakers, all working within a framework that respects privacy, promotes equity, and rewards collaboration. But here's the part most people get wrong. They think solving Alzheimer's is solely a matter of finding the next drug or developing a new scanner. The real breakthrough lies in rethinking the entire ecosystem of research and care. By embracing the gut-brain connection, by turning a stool sample into a predictive,